Okay, hi everybody. My name's Mara Carroll, and I'm the Development Officer here at Jet Foundation. Thank you so much for joining us today. We are going to get started in just about a minute. We're so glad we have Dyne Therapeutics here today joining us. Excited you're all here, so we'll wait just 1 minute and then we'll get started. Thank you. Okay, I think we're ready to get started. Hi, everybody. My name's Mara Carroll. I'm the Development Officer here at Jet Foundation. Thank you so much for joining our community webinar series today. We are so excited to have Dyne Therapeutics joining us today to talk about their Duchenne program. While we really miss seeing you all in person, we're really grateful to have these opportunities virtually. We hear from our families all the time that they're very convenient and easily accessible. So really grateful that you're all here with us today. Before we get started, I just wanted to mention that this is being recorded and will be saved in our recorded library. So if you have to leave or can't stay for the whole presentation, you will be able to watch it later. And I think, and also towards the end, it's about a 30-minute presentation. And at the end, we will have plenty of time for question and answers. So there is a question box on your screen. So feel free to ask any questions throughout the presentation, but we will get to them at the very end. So with that being said, again, Dyne Therapeutics here today. We're excited to have Ash Dugar coming with us today. I know they recently published some preclinical data that's fresh off the presses, and this is their first webinar that they're going to be sharing some of that with. So anyway, without any further ado, we'll take it away, Ash.
Great, thank you, Maura. All right, let me see if I can get this going here. Wonderful. Hopefully everybody can see my title slide up here. So, first of all, as was mentioned, I'm Ash Dugar, the Global Head of Medical Affairs at Dyne Therapeutics. I really want to thank the Jet Foundation for inviting me to present. I've been privileged to be invited by the Jet Foundation over the years and witness the extraordinary work they've done for the community. And it's really been a great learning experience for me, from the moment that I started in working in Duchenne some years ago, and I've learned a great deal from the foundation and from many members of the foundation. So I'm thrilled to be here. It's a privilege. So thank you for inviting me. Today I'm going to talk about our FORCE platform, and I'll talk about the newest data from our Duchenne program and also our clinical development plans. These are our forward-looking statements that I have to put up. It just states that I'll be making statements that involve risks and uncertainties. So at Dyne, it all starts with our mission. We're laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is truly core to who we are and what we do every day. Over the years, we've created a multifaceted program involving community webinars, conference presentations, fireside chats, and other outreach to raise visibility and gather input to advance our drug development efforts. Our head of patient advocacy, Molly White, has been absolutely instrumental in providing this foundation for Dyne Therapeutics. Over the past few years, we've been validating our platform and technology, including our ability to target delivery of potential therapeutics to muscle. And we've generated very exciting preclinical data across multiple indications and disease models. And our focus today is on sharing our newest data from our Duchenne program. So On this slide, it shows you the 4 pillars that we're using as a foundation for Dyne. We are building Dyne as the leading muscle disease company to deliver for families with Duchenne and other muscle diseases. Shown here are the 4 core strengths that help us deliver on that. The first is that we've intentionally designed what we call our FORCE platform to overcome the current limitations of muscle tissue delivery with modern oligonucleotides. The goal for our DMD program is to develop a therapy that will robustly deliver our exon skipping therapy to cardiac and skeletal muscle, including diaphragm, leading to substantial increases in dystrophin expression with relatively infrequent dosing to modify disease progression. The second is our focus on rare muscle diseases. We've generated very exciting preclinical data that shows the power of our technology across diseases and in Duchenne in particular. And I'll highlight some of that data today. The third is making sure that we have robust, thoughtful, patient-centric clinical plans in place. And so we are looking to develop our initial therapy in Duchenne for 51 skip-amenable patients and then expand from there. And we are completing the required studies right now to prepare for clinical trials by the middle of next year. And finally, I would emphasize that we have exceptional in-house and advisory expertise with deep knowledge of Duchenne and novel therapeutics, with a world-class scientific advisory board, which includes luminaries such as Dr. Francesco Montoni and Dr. Lou Kunkel. This slide provides an overview of Duchenne, which you all know very well. I don't need to go through it. Our goal is to develop a best-in-class targeted exon skipping treatment, as I said before, that substantially increases dystrophin expression with less frequent dosing than current agents, with the hope of potentially stopping or reversing the disease. I'm going to linger on this slide for a little bit. I think this is really the foundation of the FORCE platform. So our FORCE platform consists of a specific delivery mechanism called a Fab, or an antigen binding fragment, that we've engineered to bind to a receptor called TFR1, transferrin receptor 1, that is found across the surface of muscle cells, including cardiac, skeletal, and smooth muscles. So it's highly expressed across all of these muscles. Now, if you look at the Y-shaped structure on the left part of the slide, that's how a full antibody or a monoclonal antibody looks. Our FORCE platform uses the top piece, the Fab, which binds very tightly to this TFR1 receptor on muscle cell walls. And we believe Fabs have several advantages over these full antibodies or these monoclonal antibodies, including their much smaller size, which helps with distribution to the muscle tissue. Again, the FORCE platform is designed to be able to drive more of the oligonucleotide, the PMO in this case, into the muscle cells so that they can do their job that we know they do, which is exon skipping and leading to dystrophin production. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows the therapeutic oligonucleotide to enter skeletal, cardiac, and smooth muscle so that it can target the genetic cause of the disease, in this case, at the level of the nucleus. Now, we link our Fab to the potential therapy with a linker, which you see on the bottom part of the slide. The linker molecule connects the Fab to the phosphorodiamidate morpholino oligomer, or a PMO, which gets in the nucleus. In the case of Duchenne, our current potential therapy is targeted for exon 51 skip-amenable mutations. Historically, the challenge, as I've stated a couple of times, has been the inability to get enough of these PMOs into the muscle cells. And we believe our FORCE platform solves that delivery issue and can deliver the exon skipping therapy into muscle cells by attaching the PMO to the Fab with this linker, which enables targeted delivery of the PMO to muscle. So I'm going to turn to some of our newest and very exciting data. This was recently presented at Muscle Study Group Annual Scientific Congress and our recent Research and Development Day, which was held on October 13th. As an aside, all of the information, including the slides and the webcast replay, are available on our website. So on this slide, we show our FORCE platform in the MDX mouse model. It's a standard animal model in Duchenne research. These MDX mice have a mutation in exon 23. So here you can see on the top half of the slide, exon skipping in 3 different muscles, in the quadriceps, diaphragm, and heart. And on the bottom half, you can see dystrophin restoration in those same 3 muscles. And this is all over a 12-week time frame. You can see FORCE resulted in robust and durable exon skipping and dystrophin production across these key muscles with a single dose of 10 or 30 milligrams per kilogram. Now, the profound exon skipping is shown as early as week 1, and you see the highest levels of dystrophin production at week 4, but extending out to 12 weeks as well. We were excited that we saw very high levels of exon skipping in the quadriceps, diaphragm, and heart, where even at 1 week we see over 40% skipping in the quadriceps and heart, and over 30% in the diaphragm. Now, on the bottom half of the slide, I'll take you through the dystrophin data in each muscle. It stands to reason that if you're able to show robust exon skipping, then dystrophin production should follow. So indeed, at 30 milligrams of FORCE, you can see over 40% dystrophin restoration at 8 weeks in the quadriceps, and about 90% in the diaphragm and 78% in the heart, both at 4 weeks. We're extremely excited to see these levels of dystrophin restoration, especially in the diaphragm and heart, where current PMOs have had significant limitations. So on this slide, on the top panel, we show dystrophin localization. And on the bottom set of panels, we show tissue architecture. So this localization is important to see, to provide confidence in potentially seeing functional benefit. So are we actually getting the dystrophin back to the membrane so that we are confident that functional benefit will be ensuing? At 8 weeks, we see profound results, including 83 dystrophin positive fibers in the quadriceps and 80% in the diaphragm, and about 80% in the diaphragm and heart. On the bottom half, you can see restoration of the cellular architecture. And when we've shared these results with thought leaders, they share our excitement in the level of dystrophin production and PDPF that we're showing. So next, in non-human primates, another important step in generating preclinical data before going to the clinic, we evaluated Dyne 251, our lead candidate, in multiple dose regimens, as you see here. Just to quickly orient you to this study design, we looked at 2 and 4 doses of 30 milligrams per kilogram of Dyne 251 and evaluated exon skipping in this NHP model at 4 weeks. And we also administered 5 weekly doses of 30 milligrams per kilogram and then evaluated at week 8. And that's the study design in the top left corner of this slide that I just described. As a reminder, these are healthy animals, so you won't see dystrophin restoration in this model. This kind of model allows you to evaluate exon skipping and, of course, tolerability and toxicity. So the bottom line here is that with our platform, time on therapy matters. If you look at the graphs on the bottom, the 2 and 4 doses of Dyne 251 at 4 weeks are comparable in terms of exon skipping. But then when you look out to 8 weeks, we observe potentially transformative exon 51 skipping across muscles. And I, again, would point out the diaphragm and the heart, which are muscles that weaken over time, as you know. So after 5 weekly doses at 30 milligrams per kilogram, measured 8 weeks later, we saw 52% skipping in the diaphragm, 43% in the heart, and 18% in the quadriceps. And so again, we were very, very excited by this level of exon skipping in these muscles. We also evaluated Dyne 251 in a 5-week GLP toxicology study. And what we saw was a favorable safety profile in this toxicology study with no dose-limiting toxicity observed after 5 weekly doses up to a maximally feasible dose. Importantly, we pushed the dose, dosing many folds higher than 30 milligrams per kilogram, and so we're really pleased with the safety profile as we advance into the clinic. So I'm going to switch over now to a couple of slides on our clinical development plans. So as we prepare to launch our first clinical development program, we've done quite a bit of work. We've engaged with advocacy groups, patients, and key thought leaders from various disciplines, including pediatric neurology, cardiology, pulmonology, physical therapy, and adult neurology. Importantly, we've gathered this feedback across the globe. Thought leaders, key opinion leaders, both inside the US and outside of the US, with a great deal of experience running early and late stage clinical trials, have provided advice on our overall clinical development program, including our clinical trial design, the patient population, biomarker, or the patient population we should be evaluating, biomarker endpoints, functional endpoints, and of course, key safety considerations. They've also helped embed learnings from natural history studies that we should be aware of. And it's clear that these key opinion leaders believe dystrophin continues to be a key endpoint in looking at how a treatment performs in a clinical trial. We've also received advice from global advocacy leaders, patients, and caregivers that have provided insights based on their living with and being the voice of the DMD community, as well as experience with past and current trials. This input has been critical in ensuring a patient-centric design and has included key considerations for choosing a clinical trial, because there are many choices now, which is fantastic. Feedback on biopsies and functional outcomes. Identification of the most burdensome or difficult parts of participating in a clinical trial, and how to ensure patients and families are supported throughout the entire process. So based on this feedback we've received from thought leaders, patient advocacy leaders, patients and caregivers and regulators, we've developed a proposed clinical study for Dyne 251 in patients amenable to exon 51 skipping. And I'll just walk you through some of the key elements here. Our clinical trial for Dyne 251 will be a global, multiple ascending dose, placebo-controlled study with a long-term extension. We plan to enroll patients with symptomatic Duchenne DMD and mutations amenable to exon 51 skipping. Our goal is to enroll approximately 30 to 40 male participants aged 4 to 16 who are either ambulant or non-ambulant. Some of the endpoints we plan to evaluate include dystrophin measurements, measures of muscle function, both lower limb function and upper limb function, as well as respiratory function, and of course, monitoring very closely safety and tolerability. We plan to submit our IND this quarter, and just to be clear, the IND is the investigational new drug application that we will submit to the FDA. That will be the step after all our studies are done, so that we can then move into the clinic, as I said before, by the middle of next year. Now, while our initial focus is on this exon 51 program, as I mentioned earlier, our goal is to build a best-in-class pipeline to address the 80% of patients with Duchenne amenable to exon skipping. We will be looking to rapidly advance programs in exons 53, 45, and 44. The data that I've reviewed, combined with the deep engagement we've had with multiple stakeholders, has positioned us well as we make this exciting transition into the clinic. And I'm looking forward to the important work ahead as we advance our efforts to develop new potential therapeutics for patients living with Duchenne. So this slide, very importantly, recognizes just a few of the very many voices and comments that we hear. We are committed to understanding the community of people living with muscle diseases, and Duchenne in particular, and ensuring that your perspectives and your experiences are included in our drug development programs. This is central to our work each and every day, And so thank you so much for advising us and working with us. Finally, on this slide, you can see Ravi, Beth, and Diana and their families, who are all part of the Dyne family. And at Dyne, we really do make decisions with patients and families in mind. This is absolutely what drives us each and every day. Thank you.
Great. Thank you so much, Ash. That was a great presentation. To everybody that's listening, please feel free to drop any questions into the chat box below. But first, just a comment that, it's really evident that Dyne Therapeutics has really listened to the community and, in a very thoughtful way, how you've designed your program. So thank you. As you said in the beginning, you've worked very closely with advocacy for many years, and your fireside chats and things like that have been really great, and it's very evident in your program.
Yeah, Molly White, who's our head of advocacy, she is amazing. As some of you may know, she was actually CEO of the Myotonic Dystrophy Foundation for a long time, and we're very fortunate that she's part of the organization at Dyne.
Absolutely. Okay, we have a few questions coming in. First one, can a patient come off another exon skipping drug and join yours?
So we haven't finalized the protocol, so we're still working out some of the details. And when we have all of the details of the protocol worked out, we're definitely going to share it with the community. I know that's not particularly satisfying, but we're still making sure that we have all of our T's crossed, all of the I's dotted. That is a big question as well that we are being very mindful of, certainly for our clinical protocol. So more details to follow, but it will definitely be out there when we're ready.
Great. The next question is about dystrophin. Did your presentation show how much dystrophin would be assessed?
Yeah, it did. Just so we're talking about the same thing. So right now, what we have, what we've shown you, what we have is the exon skipping data and the resulting dystrophin expression from various MDX mouse studies. So there is data that I haven't shown as well that looks at these mouse studies at 2 and 4 weeks, and it's in our, again, all of that data is on our website. And in the slides that I showed earlier, just earlier in the presentation, we also show levels of dystrophin expression at various muscles. And again, within the MDX mouse model, we're getting tremendous levels of dystrophin restoration. And if you want, we can go back to the slide and I can go through it one more time, but it's definitely in the deck. And so we're really excited about the level of skipping and dystrophin expression we're seeing in the MDX mouse model.
Excellent, great.
And again, just at the risk of repeating myself, we can't do that in the non-human primate, in the monkey model, because they're healthy. There is no dystrophic monkey model, so you actually cannot look at dystrophin restoration in the non-human primate.
Great. The next question is, I guess, when you begin enrolling patients in your trial, will you be providing travel, food, and other assistance?
So the current plan is to make sure that we have really taken into account what the patients and families need to be able to not bear costs, with their participation in the trial, that we can actually help them manage. That we're also making sure that we are putting together a plan so that every family knows exactly what to expect, the moment, even before they walk into the door for a clinical trial, what to expect during that day, and then what we can do during that whole experience to make it as easy as possible, because these are such complicated protocols and quite a lot of burden. So I just want to sort of start with that general statement that we're trying to be mindful of everything that we can think of and everything we're getting in terms of feedback from patients and caregivers in terms of what they've experienced, both positive and maybe less positive, in participating in clinical trials. So we're trying to be mindful of that. To your question on costs, the answer is yes. I mean, currently what we're doing is planning to make sure that we have reimbursement services in place or other strategies like prepaid debit cards or something like that to be able to minimize that financial burden on the families.
Great, thank you. Do you have yet, maybe this, you don't have it just yet, but do you have a list of site locations you're planning on?
Yeah, so we don't at this time. Again, as soon as we're ready to announce it, we absolutely will announce it. We're still going through the processes, as you can imagine. We're planning to start the trials in the middle of next year, so we're still going through the process of identifying sites, engaging with sites, etc. So that information will be coming. We're just not ready at this time.
Okay, the next question says, what limits the percent of skipping to 20 to 40%? Why wouldn't all mRNA be impacted?
Well, it is a good question, and I don't know that I'll give you a perfect answer here. I'm not sure that there is a perfect answer, to be honest with you. So historically, the big limitation in being able to get to, I won't even say 20, to get to like 4 or 5, 6% skipping, is getting enough of the morpholino of that oligonucleotide into muscles, and that has been the biggest challenge, is being able to drive the PMO into muscle. Then there's going to be uptake into the cell, into the nucleus, and so it is unlikely that every single cell will have the ability to drive or absorb that naked or unconjugated exon skipping drug, if you will. And then what has been shown then, subsequently what's been shown in the clinic, is very limited dystrophin production with some of the current therapies. They clearly work, right? They are able to do what they're supposed to do in terms of exon skipping, and then have some but limited dystrophin restoration. So what we're trying to do is really use that Fab, attached to the PMO, using the transferrin receptor to drive more of the PMO into the cells. And so what we're able to show in the preclinical models is the ability to have much more PMO driven into the muscle cells for much more skipping and dystrophin production.
Great. The next question is, will you be considering home infusions for this study?
Yeah, we've gotten that question. So the answer is we are considering it, and again, that is part of some of the operational aspects, one of many that we need to still figure out for our clinical trial. But we absolutely know from experience, from other clinical trials, but also experience with patients that are on commercially available exon skipping drugs, the importance of home infusion. So we are acutely aware of it and want to figure out how to make it work for trials. I will say that the other aspect, I think related aspect to that, is we will not be dosing weekly. We have generated data that gives us confidence that we can have much less frequent dosing than weekly dosing, perhaps monthly dosing. And so, although I still think home infusion is important, the good thing is it won't be weekly dosing. most likely monthly.
That's great. Next question, will you test your therapy in patients who have received microdystrophin gene therapy?
So it's a great question. We have gotten a lot of questions around would we allow patients, I think to the earlier point, with prior eteplirsen use, gene therapy, with with other symptomatic treatments like vamorolone or others, right? Like we've had a lot of those kinds of questions, and I guess I'll answer maybe the bigger question. So again, we're still grappling with some of these questions, and we have a very good perspective on what we need to do, but we're still working out some of those details. On the other side, I don't want to hypothesize what other sponsors may or may not choose to do with what they may allow if patients are on gene therapy clinical trials. But there's also that aspect, right? So for example, sponsor X, who has patients enrolled for a gene therapy study in Duchenne, they may not allow that patient to go on another investigational drug while they're in their study.
Okay, next question is, will your protocol require being on a steroid?
Yeah, so I think that we're very likely going to require steroid use. What feedback we've gotten from the physician community is that within the United States and most parts of the world, that is standard of care, and so to truly understand the full treatment effect of Dyne 251, it probably makes sense to administer it on top of patients who are on steroids. And so there are just a couple of countries that I'm aware of, at least in the world, where there's more variable use of steroids, or where it may not be considered standard of care. So it does make a lot of sense. And again, that's another detail that we're going to be finalizing and end up communicating to the communicating.
The next question is, will this study require a biopsy?
So if we do believe, which again is the feedback we've gotten from the physician community, that dystrophin expression is an absolutely essential endpoint for this type of study, for an exon skipping study, where you are producing a shortened but functional form of dystrophin, that a biopsy would be needed to be able to understand where is the person living with Duchenne at sort of time zero, and then how has that dystrophin expression changed over time. So it's very likely that we're going to require a biopsy. And I think, yeah, I'm sorry, go ahead, please.
No, I was going to say, I don't see any further questions coming in. So if you had any final comments, please feel free.
Yeah, thank you. First, again, if there's any questions that the community has, you can reach out to me and get my contact information through Maura Molly's as well, and we will get back to you. We may not have complete answers for you, or any answers in a few cases, but we will definitely get back to you. We're committed to communicating information when we're ready to communicate it and make sure that it's solid, and it's going to be sort of what our perspective is and moving forward. We have newsletters that we provided to Jed, and there's a bunch of information on our website. So we want to be able to provide as much information as possible. Second, I'll just reiterate our excitement over the FORCE platform. We think that this really has tremendous potential to solve what I think is one of the, if not the biggest issue with current exon skipping drugs, which is trying to deliver the morpholino to muscle in a way where you get very, very robust skipping and dystrophin production, so that you can actually truly change the course of disease. And then finally, I just want to thank you again. Jeff Foundation has been just an organization that has educated me over the years, truly educated me over the years. I came in, I didn't know, years ago into Duchenne, and I didn't know much about the disease. And it was some of the leaders and families at Jet Foundation that helped me understand truly like what this is, beyond the science and beyond the medicine, like what are families going through. And so thank you for this opportunity, and I do find it to be a privilege to speak to you today.
Great, thank you so much, Ash. And we too, as an advocacy organization, are really grateful to Dyne for bringing these webinars to us so early, for engaging with advocacy so early, and being part of all your protocols. It means a lot, and we know that exon 51 is a competitive landscape, and you let us know too what we can be doing to help, because it sounds like this is a great program. So for everybody joining us today, thank you for being here. Again, this is a webinar that has been recorded, so we'll be sharing that on our website very soon. And if you have any questions that you didn't think of while you were listening, like Ash said, you can reach out to me, and I'll get it to Molly and Ash, and look forward to hearing all that unfolds with you and your program. Thank you so much, Ash.
Great, thank you, Maura. Thank you, everyone.
Have a great afternoon. Bye-bye.
Bye.